As late as the 1930s, biologists still did not understand what genes were or what they did. Today, we realize that they are segments of DNA, largely because of work in the 1930s by George Beadle and Edward Tatum of Stanford University. We now know that one of the most important things genes do is to store information for the creation of proteins. In particular, they store information for the creation of enzymes, the proteins that hasten reactions. For a number of years, Beadle and Tatum studied the red fungus Neurospora, a bread mold. In its natural or wild-type state, Neurospora can manufacture most of the chemicals it needs to live and grow, provided it has access to a few simple biological substances. A minimal set of these substances is called a minimal medium. The researchers induced mutations in the Neurospora by irradiating spores of the fungus with X-rays. They then placed the mutated species in containers with liquid, having the minimal set of biological substances that was sufficient for the wild-type Neurospora to survive. Beadle and Tatum now found that the mutated strains of fungus were unable to grow in the minimal medium. Although they had access to the same basic raw materials used by the wild type, the irradiated fungi were apparently no longer able to produce some of the substances they needed to survive. Beadle and Tatum then added different substances to different samples of the irradiated spores in minimal medium. In most cases, the mutated fungi still could not grow, but in some cases they could. In this way, the scientists were able to determine the substances that the wild type could make, but that the irradiated fungi could not. In fact, they found that different samples of irradiated spores required the addition of different substances. In some cases, the substance that had to be added was the amino acid arginine. We now know that arginine is produced by Neurospora in a metabolic pathway, a chain of chemical reactions controlled by enzymes where the product of one reaction serves as the substrate, the substance on which an enzyme acts, for the next reaction in the chain. In this chain of reactions, a chemical known as a precursor is modified over several steps, with a different enzyme acting at each step. By selectively supplying different mutant strains with different precursors of arginine, it was possible to discern the step at which the arginine metabolic pathway had been blocked by the mutation. They found the pathway was blocked at different points for different mutant strains. For instance, one strain required only precursor A to be able to produce arginine, while another required both precursors A and B. They concluded that a given mutant had been mutated by having its genes altered by the X-rays in such a way that the strain could no longer transform one precursor molecule into another. Since the addition of the enzyme to the minimal medium was required for the production of arginine, Beadle and Tatum argued that what genes were doing was producing enzymes. Eventually, the conclusion of these experiments was popularly summarized as the one gene, one enzyme hypothesis. That is, the role of a particular gene is to produce one enzyme that has a particular metabolic task. Subsequent research has modified details of the one gene, one enzyme hypothesis. For example, a more correct statement is that a gene codes for a polypeptide, which is a component of a protein, which is in turn a component of an enzyme. But the essential insight of the hypothesis, that the primary role of a gene is to produce a specific protein or enzyme product, was critical to the understanding of what genes do. In modified form, this insight is a central precept of modern biology.